I always laugh. Can you hear me? Or hello? Okay. Okay, hello everybody. Today we are going to be going over some material for lecture exam 4. Oh, hey Laura, can you hit the thing again? Yeah. Let me make sure it works. I don't know why it does that. Oh, I did that. Okay, there we go. So lecture exam 4 goes over 3 chapters. It's going to be chapter 11, Fundamentals of the Nervous System and Nervous Tissue, chapter 12, the Central Nervous System, and then chapter 13, Peripheral Nervous System and Reflex Activity. Chapter 12 is a lot of anatomy, so that's a lot more lab-based, and today we're only covering lecture stuff. So with chapter 12, when you are reading through it, you need to know a lot of the structures by sight, but more specifically for the lecture, you're going to want to know the specific parts or functions that those structures do. And then we'll talk a little bit about that. My class today, they should be testing on Wednesday. We only finished chapter 11. So I know, at least for one professor, Mr. Torres, he's going to be sending out a review type thing that he did for a previous class. Just basically going over exactly what you'll need to know for the exam, since we didn't get too far in the chapters that were going to be covered. So we're starting with chapter 11, functions of the nervous system. The big 3 things with the nervous system is sensory input, integration, and then motor output. So sensory input is basically using your senses, seeing, hearing, tasting, things like that. And then sensory input is information gathered by your sensory receptors about internal or external changes. So we can see here in the image, we are using our sense of sight to see the water. From there, we go to integration, which it usually occurs in, or it does occur in our brain. This is when we start processing or interpreting the sensory input. So this is when we realize we see the water, we think, oh, it's been a while since I've had something to drink, I'm kind of thirsty now. And then we have a motor output, which is the activation of effector organs. And then that produces, that should say a output. So we do something with it. So we're using the motor output or our muscle to pick up the water and then take a drink. So this is the big overview of the nervous system. Now things get a lot more complex, obviously, but this is like the basic what our nervous system does. We have a sensory input, integration, and then motor output. Okay. Oh, this one. And I got it. Okay, and then, so this is getting into the specific parts of chapter 11. So the big things to know here is that we have 2 divisions of our nervous system. The central nervous system consists of our brain and spinal cord. And again, we call this the integration or control center. And again, that's our brain. So central nervous system, brain, and spinal cord. And then we have our peripheral nervous system, which are the nerves and ganglia, and these are outside of our central nervous system. So our central nervous system, again, is more like the midline of our body, brain and spinal cord. Peripheral is everything else. With the peripheral, it gets a little more complex because it has 2 subdivisions. That's the sensory or afferent, and then motor or efferent. However, that helps you to memorize it, efferent or efferent. I say efferent, but just depends on how it helps you remember it. And then within the motor system, we have 2 subdivisions of that division, so somatic and then autonomic. So again, central nervous system, brain and spinal cord, peripheral nervous system, sensory and motor, and then the motor has 2 subdivisions, somatic and then automatic. So sensory or afferent, these have sensory or afferent also have different types of fibers. So we have somatic sensory fibers. These convey impulses from the skin, skeletal muscles, and joints to the central nervous system, and these are voluntary. So big thing to know here, somatic, voluntary. Again, so this would be picking up the glass of water because you're thirsty. Your visceral sensory fibers convey impulses from the visceral organs to the central nervous system. This is involuntary. So again, the difference between somatic and visceral, one is voluntary and then the other is involuntary. And then with our motor or efferent, this transmits impulses from your central nervous system to your effector organs. So with the sensory, these are conveying impulses to the central nervous system, whereas with the motor or efferent, they're transmitting from the central nervous system. So that's another big thing to know, which is conveying impulses to and which is conveying impulses from. I would make sure you know that for your exam, they can ask you, or they can give you the definition for one of these subdivisions and then ask you to identify which they are explaining. Make sure I got everything. Okay, so with nervous tissue, there's a lot to know. A lot of this for lecture, you need to be able to identify which type are like most abundant, which are found where, whether it's the central nervous system or the PNS. And then for the lab, you're going to want to be able to actually identify them by site. That is on your lab worksheets. So we have two principal cell types of nervous tissue, neuroglia, and then the neurons. Neuroglia are small cells that surround and wrap the delicate neurons. And then the neurons or nerve cells are excitable cells that transmit electrical signals. So you can see on here, I have them in different colors. So I have 4 that are in blue and then 2 that are in purple. Do we know why that is? Exactly. So the ones in blue are found in the central nervous system. And then in purple, these are found in your PNS. So let's go through them. Astrocytes, these are the most abundant. And again, they are found in the central nervous system. These cling to neurons, synaptic endings, and capillaries. And then these support and brace neurons. And then they have an exchange, or they play a role in exchange between blood capillaries and neurons. The next one are the microglial cells. These migrate toward injured neurons, and these are described as like the defensive cells. These are also the phagocytes. Again, CNS, the ependymal cells line the cavities of the brain and spinal column and then form a barrier between CSF-filled central nervous system cavities. What is CSF? Anybody? Yes. So CSF, anytime you see that cerebrospinal fluid, so the ependymal cells are found in the barrier between your cerebrospinal fluid and the cavities. And then the oligodendrocytes, these are, again, in the CNS. These form the myelin sheaths. And that is on our big neuron model that we have. And then getting into the PNS, satellite cells, these are similar to astrocytes. These surround the neuron cell bodies, and do we know where? Axons, yeah. And then the Schwann cells, these are kind of like the oligodendrocytes in that they produce or form myelin. The only thing with these is that these are found in the peripheral nervous system. And let's see, I have a picture of a few of them here. So this first one is the astrocytes, the one at the very top up here. Again, these are more so you need to know for lab to identify them by sight. But for the lecture, you're going to want to know the function of each of these and then if they're CNS or PNS. This specific image is from your textbook. So if you're looking for this image, it's in your textbook, chapter 11. Okay, then I just have a bunch of definitions for like the anatomy of the neuron. These, again, are in your book, and they're important to know for lecture. So I'm going to go through them pretty quickly because I know it's a lot. First, we have the types of neurons. So we classify them in functional and structural. So we have for functional, sensory, motor, and then interneurons. And then structural, we have multipolar, bipolar, and unipolar. From the types of neurons, where are multipolar neurons found? Or where are they most abundant in which system, which division of the nervous system? Good guess, they're in the central. Yeah. So with that, where do we think the unipolar are mostly found? If multipolar was central, where do we think unipolar is? Peripheral, yeah. So multipolar, mostly found in the central. Unipolar, mostly found in the peripheral. Multipolar is exactly what it sounds like. They have many processes, and then they are the most abundant. Bipolar have 2 processes, and then unipolar have just one single process. For lecture, again, I would know the definition of them, their characteristics, so many, little, or 2, and then where they are found. Okay, so getting into the anatomy of the neurons. So neurons, again, the structural unit of the nervous system. We have our neuron cell body, or again, we call it the soma. This is the biosynthetic center of the neuron. It synthesizes the proteins, membranes, etc. This is one of the more important things to know. And then we have the nucleus. It's a collection of neuron cell bodies in the central nervous system. So when we're talking about the central nervous system, we'll be talking about the nucleus. The ganglion or ganglia are the collection of neuron cell bodies in the peripheral nervous system. So again, peripheral nervous system is everything outside of the CNS. That's when we have our ganglion. A tract is a bundle of axons in the CNS. And then our nerves are bundles of axons in the peripheral nervous system. Our dendrites, these receive input or signals from other neurons. They convey messages toward the cell body as graded potentials. So these are the short distance signals. Axons, these generate nerve impulses, and then they transmit along the axolemma to the axon terminal. Molecules in organelles are moved along the axons by motor proteins and cytoskeletal elements. And then we have the anterogrades and retrogrades. Anterograde just means it's moving away from the cell body, and then retrograde toward the cell body. So anterograde, away, retrograde, toward the cell body. My professor that I sit in on, he kind of stressed this, so I would make sure you know these 2 definitions right here, anterograde and retrograde. And then lastly, we have the myelin sheath. These are composed of myelin, protects and electrically insulates the axon, and then it increases the speed of nerve impulse transmission. I think that says it moves slower. Conduct. Yeah. So when the sheath is non-myelinated, the fibers conduct the impulses slower. So it does not move as quick as the myelin sheath does. It moves slower. So it'll take barely any longer than it normally does. But when it has that characteristic of being non-myelinated, it does travel slower. Oh. Let's see. Let's make sure I got everything. Okay. And then we get into action potential. This is some things that a lot of students have trouble with just because it is a little bit more complex. The way that I like to learn things is I like to find the most elementary video I can find. So something that really breaks it down as simple as it can be. And then I like to read up on it more or watch a more complex video. So if you're having trouble with action potential, that's something that I would suggest you do is find a really elementary or easy video and then work your way from there. So action potential is a nerve impulse. It's a brief reversal of the membrane potential that occurs in axons and then results in the release of a neurotransmitter. And so again, action potential is the principal way neurons send signals over long distances. The caveat with this is that only cells with excitable membranes can generate action potentials. So if there are no neurons or muscle cells, there is no action potential to be created. And then the action potential begins and ends at resting membrane potential. This is depolarization to about 30 millivolts is caused by influx. And then the depolarization ends when the channels inactivate. So again, depolarization is caused by influx. We get repolarization and hyperpolarization when we have efflux. If our threshold is reached, then an action potential is generated. If not, then the depolarization remains local. So when we talk about being local or staying local, it's restricted to the area of the dendrite in which the stimulus was applied. So we have three types of like local graded potentials. We have, like I said, local, restricted to the area of the dendrite in which the stimulus was applied. Graded potential is the change in the membrane of the dendrite, is in proportion to the strength of the stimulus. So the stronger the stimulus, the more ion gates open. And then potential is the opening of ion gates results in the flow of ions across the cell membrane and results in a change in charge. Okay. And then in nerve impulse propagation, each action potential provides the depolarizing stimulus for triggering an action potential in the next patch. Regions that have just generated action potentials are refractory. And then for this reason, the nerve impulse propagates in one direction only. And then action potentials are independent of the strength of the stimulus. A strong stimulus causes action potentials to be generated more frequently, but not with greater amplitude. And then during the absolute refractory period, a neuron cannot respond to another stimulus because it is already generating an action potential.
Then when it's repolarizing, it's like on its way like back down to the
Yes.
and then not absolute, the other one that's
Refractory?
refractory. Is that when it is hyperpolarization?
Yes.
That's where it's going back to its normal state.
Yeah.
I'm having a
Yes.
hard time understanding how it works.
Yeah, these are confusing, or it's a lot to get. But yes. Okay, so then we have our image here, which I know again isn't very large, or you can really see it. This is again in the textbook. It's just showing you right here. We are starting at number 1. It says resting state. 2 right here is depolarization, repolarization, and then hyper down here. So we have our resting state, depolarization, repolarization, and then our hyperpolarization. Again, this is in your textbook, chapter 11. I don't have the actual figure number, but it is in chapter 11. Another thing I would know is the voltage. So making sure you're knowing the numbers that occur at each. So at the resting state, it's about -70 millivolts. I couldn't think of it. Or are we next? Oh, depolarization, it's about +30 to 40 millivolts. Repolarization, it's a little bit more than that one. And then hyperpolarization is when it gets really low. And then right here is our threshold. So remember, the threshold has to be reached in order for the action potential to be generated. If it's not reached, then the depolarization remains local. And then another big thing to know, action potentials are all or none. So again, if the threshold is not reached, nothing occurs. Oh, I forgot about that. Okay. And then chapter 12, again, this is a lot of brain anatomy. So a lot of this is covered in lab. Today, what we're really going to go over is the specific structures that have specific functions for your body, but a lot of this you will cover in lab. So the first thing is EBD, or early brain development. The structures, this is the prosencephalon, which are your cerebral hemispheres, and the diencephalon. The mesencephalon, which is your midbrain. And then your rhombencephalon, which are your pons, medulla oblongata, and cerebellum. Again, for lab, you're going to want to know the specific regions and where they belong. Again, this is early brain development. Okay, I know this is a lot. I'm going to break it down. So your adult brain is divided into the cerebral hemispheres, the diencephalon, brainstem, and cerebellum. When we are talking about brains in general, we're usually always talking about a fully developed adult brain. I know in lab we dissected sheep brains. Those may come up on the exam, but when we're asking about specific functions or identification, we're usually talking about an adult brain because all of the pieces are there and working. So again, adult brain divided into cerebral hemispheres, diencephalon, brainstem, and cerebellum. The brain has 4 ventricles, which are filled with cerebrospinal fluid. These are the lateral ventricles, your third ventricle, and the fourth ventricle. So I just named 3. Why did I say that there's 4 if I named 3? Remember, it's the lateral ventricles, third ventricle, and fourth ventricle.
Like one of
Yeah, you have 2 lateral ventricles, so they're like this. Okay, and then each cerebral hemisphere receives sensory impulses from and dispatches motor impulses to the opposite side of the body. So you've ever heard like the left side of your brain controls the right side of your body and vice versa. So we have specific areas of the brain that you're responsible for knowing for the lecture portion. The motor areas, this is the primary motor and premotor cortices of the frontal lobe, the frontal eye field, and the Broca's area. The Broca's area is found in the frontal lobe of one hemisphere. Do we know what Broca's area is, or okay, take it back. Do we know what damage to the Broca's area does to like your body? What function kind of goes away? Speech. So damage to the Broca's area involves or can result in a delay or you no longer being able to speak. Oop. We talked about the premotor cortex. This controls learned motor skills and repeated patterns. So damage to the premotor cortex is leads to a loss of learned skill. So if you knew how to play the piano, you would have to relearn it. Mm-hmm. Yeah. Okay, and then we get into the sensory areas. These are the primary somatosensory cortex and the somatosensory association cortex in the parietal lobe. There's a few of these. The visual areas that are in the occipital lobe, olfactory and auditory areas in the temporal lobe, gustatory, visceral, and vestibular areas in the insula, so the sensory area, like I said, what do we think these specific structures in our brain are responsible for, or what do they play a part in? If this is called the sensory area. Senses, yeah. So seeing, hearing, tasting, things like that. So we can put two and two together that the visual areas in the occipital lobe, damage to hear would affect what? Our vision. What about damage to our temporal lobe right here? If the olfactory and auditory areas in the temporal lobe, say we took a big hit or we had some damage to our temporal lobe, what would be affected or what could possibly be affected? Hearing, yeah. Okay. And then we have association areas. These are found in the frontal lobe, in the anterior part of our frontal lobe, and the posterior and limbic association areas span several different lobes. So this is where we find like our Wernicke's area, so the language area. What else? Our wait, where are we? Spanning several. I would know the specific parts that are in each of these areas. So what is in the motor area, what is in the sensory area, and then what are in the association areas. I don't remember if I have a picture. No, I don't. Okay. Again, a lot of this is in the lab where you'll actually go over the specific parts of the brain, but you do need to know the functions of those parts for lecture. Okay, so then we get into our diencephalon, which is here on the right side, this image here. This is purple part right here is when the brain is whole and put together. This is a cut of the brain. And it's this tiny little purple area here. We call this the diencephalon. This has the thalamus, hypothalamus, epithalamus, and it encloses the third ventricle. So again, what are the ventricles, or what is going through the ventricles? CSF, cerebrospinal fluid. First, we have our thalamus. This is the major relay station for sensory impulses. It gets inputs from the subcortical motor nuclei and the cerebellum traveling to the cerebral motor cortex. And impulses traveling to association cortices from the lower centers. So here, again, is the diencephalon. This is it whole. This is it cut. The thalamus is also the largest part of the diencephalon. It receives input, edits, sorts, and relays to appropriate cerebral cortex areas. So it's kind of like the messenger. So we have our thalamus. Our hypothalamus is an important control center of the autonomic nervous system and our limbic system. It maintains water balance and regulates thirst, eating behavior, gastrointestinal activity, body temperature, and the activity of the anterior pituitary gland, which is down here. So there is the pituitary gland, and the hypothalamus controls the activity. Another big thing is the hypothalamus maintains homeostasis. So I would make sure you know that for your exam. It's responsible for, again, regulation of body temperature, food intake, thirst, sleep-wake cycles. And then our epithalamus, this is our pineal gland, or it is within, kind of near our pineal gland. What does the pineal gland secrete? Does anybody remember? It's a specific hormone that helps us do something that we need to do every single day. Melatonin, yeah. So pineal gland, epithalamus. So if they were to ask which gland is found in the epithalamus, pineal gland, what does it do? Secretes melatonin. I think that's really what you need to know. Again, big thing to know, hypothalamus maintains homeostasis for your body. Hypothalamus, yes. Okay, getting into some more structures. Here we have our brain. This should be a, if it were like upside down, so inferior view. Spinal cord. Right now we're going to look at the midbrain. So that is here. Trying to think what else from here. Yeah, okay. So the midbrain contains bundles of nerve fibers, and it joins the lower brainstem and the spinal cord with higher parts of the brain. It has the cerebral peduncles I hate this word, corpora quadrigemina, and the substantia nigra. Do we remember the name for like the overall midbrain? It's one of those longer ones, starts with an M. It's the mesencephalon. Within the midbrain, there's several masses of gray matter that serve as reflex centers. And then also within your midbrain is the cerebral aqueduct. Let's see. The corpora quadrigemina is, or they include centers for visual reflexes and auditory reflexes. See it now. But again, that's found within the midbrain. That's the corpora quadrigemina, not Gemini. Visual reflexes and auditory reflexes. And then moving down, we have the pons. The pons are a lot easier to see when you have a model of a brain. So when it's cut in half. So if you have time in the lab or in here when we finish, go ahead and look at one of the brain models so you can see the pons. The pons relay impulses to and from the medulla oblongata and the cerebrum. So the pons are in here. They relay messages to the medulla oblongata and the cerebellum. Sorry, not cerebellum, cerebrum. And then the medulla oblongata. This is the enlarged continuation of the spinal cord. Who remembers what the large hole at on the bottom of the skull is called? The large hole. So if we turn the skull upside down, there's a really big hole. Starts with an F. The foramen magnum. So the medulla oblongata, again, if we're thinking of where the brain is located inside of our skull, it's the continuation of the spinal cord extending from the pons to the foramen magnum. And this is the most inferior part of the brain. Okay, and then within the medulla oblongata, we have 3 centers: the cardiac, vasomotor, and respiratory center. What do we think the cardiac center, which organ does that work with? It's not a trick question. The heart, yes. So the cardiac center of the medulla oblongata transmits impulses to the heart via peripheral nerves, and this causes your heart rate to increase or decrease. The vasomotor center transmits impulses to smooth muscles in the walls of blood vessels, causing them to contract and constrict. This is what we call vasoconstriction. And then there's also cells that oppose the constriction. So this is the dilation of blood vessels, and this is vasodilation. So again, cardiac center transmits impulses to the heart, causing heart rate to increase or decrease. The vasomotor center has vasoconstriction and then vasodilation. And then the respiratory center functions with the pons, which we just talked about. We can see them here, pons, medulla, to regulate the rate, rhythm, and depth of your breathing. So again, these 3 centers are within the medulla oblongata. And again, if we were to, I don't think I have a picture. I don't. If we were to cut it sagittally and we opened up the brain, we would see the pons kind of hanging out and attached to the medulla. And that's why they work together. They're very close together. We just cannot see it in this picture because this brain is whole. And then our cerebellum, which are down here, right here. And then on this side, we have two. Processes and interprets impulses from the motor cortex and sensory pathways and coordinates motor activity so that smooth, well-timed movements occur. So the cerebellum receives sensory impulses from receptors in muscles, tendons, and joints. And then so because of that, it serves as a reflex center for muscles. Okay, this again is a little bit more lab, but I wanted to put it in here because my professor talks a lot about it in class, so I can see it coming up on your lecture exam. These are the meninges of the brain. So we are starting on the very outside, kind of what we can see. Here is your scalp. Periosteum, do we remember what that is? That's that outside like covering of bone. So if we look at the bone model that's back here, that's this portion. So it's the covering of bone. We also have that underneath our scalp because of our skull. So our skull, periosteum, which is the bone covering, and then our scalp. Right underneath our skull, we have the hardest and also like thickest layer of meninge, which is the dura mater. It's the outermost membrane, and it has many blood vessels and nerves. If you have not gone to lab yet, or you can do a sheep dissection. You can pull back on the dura mater and it'll like snap back at you because it's really thick, because it is pretty much like that first layer of protection for your brain, because we're going from skull straight to the dura mater. That's why anything with the head, any sort of injury with the head is really important and is to be taken very seriously, because this is all that is protecting your brain. And then we have, let's see, right here in between our skull and the dura mater, we have the epidural space. So bone of skull, dura mater, right here, epidural space. There is blood vessels, loose connective tissue, and fat to provide padding. And then we have a few layers of the dura mater. We didn't go over these, so I don't think you need to know these. The second type of meninge is the arachnoid mater. The important thing here is underneath the arachnoid mater is the subarachnoid space. The subarachnoid space is important because it contains cerebrospinal fluid. And then after the arachnoid mater is the pia mater. The pia mater is the innermost, and then it's also very thin. So that's kind of the first layer of protection with your brain because it directly covers it. So the way that I always remember the order of these is brain and then pad. So you have your brain, then you have pia mater, arachnoid mater, dura mater, and then your skull. So brain, pad, skull. If we were working our way from the outside in, it would be skull, dura mater, arachnoid mater, pia mater, brain. Again, big thing to know, where'd it go? Oh, the subarachnoid space, cerebrospinal fluid is found in here. And then we have some of the different structures of the brain. So the limbic system consists of numerous structures that encircle the brain stem. This is the emotional visceral brain, and it plays a role in memory. So again, limbic system, memory. Let's see. In most people, the left hemisphere controls language. The language implementation system is the Broca's and Wernicke's area and the basal nuclei. The basal nuclei analyzes incoming and produces outgoing language. And then the opposite hemisphere deals with the nonverbal emotional content of language. So left hemisphere controls language, the language implementation system, the Broca's and Wernicke's area. And then another definition, memory is the storage and retrieval of information. It is essential for learning and it's part of consciousness. There's two stages to memory, short-term memory and long-term memory. Short-term memory is processed in the frontal lobe and thalamus. And then, oh my gosh, long-term memory, it is in the temporal lobe, sorry. Okay, then we get into the peripheral nervous system. So we talked about it a little bit earlier, the central nervous system. Peripheral nervous system has a lot more kind of subdivisions. So we have the sensory or afferent, motor efferent division. Within the motor division, there's somatic and then autonomic. And then within the autonomic is sympathetic and parasympathetic. So my class did not get this far, so I'm not entirely sure how much of the chapter you 100% need to know, but I'm just going to go over kind of like the overview of chapter 13. So this is talking about sensory receptors. These include the receptors for your general senses, pain, touch, pressure, and temperature. Receptors in the skin, as well as receptors in skeletal muscles and tendons and in the visceral organs. Sensory organs contain sensory receptors and other cells that serve these special senses. Again, these are eyes, ear, taste buds, olfactory, things like that. Okay, this is a lot. So receptors are classified according to the stimulus. So we have mechanoreceptors, thermoreceptors, photoreceptors, chemoreceptors, and nociceptors. And then they're also classified according to its location. So we have exteroceptors, interoceptors, and proprioceptors. And then once you break these down, they're kind of common sense. So thermoreceptors, temperature, photoreceptors, sight, chemoreceptors, chemical things that are happening in the body, mechanical. Okay. So again, let's go through that. Mechanoreceptors, sensitive to mechanical forces, so pressure or movement of fluids. Chemoreceptors, sensitive to changes in concentration of chemical substances. Photoreceptors, sensitive to changes in light intensity. Nociceptors are classified as pain sensitivity. The nociceptors can be as a result of chemical, mechanical, or temperature extremes. And then thermoreceptors sensitive to changes in temperature, so heat and cold. Oh, they're here. And then we said they're also classified according to location. So exteroceptors, these respond to stimuli arising outside of your body. So where we touch, pressure, pain, and these are most of our special sense organs. So most of our special sense organs fall under exteroceptors. Interoceptors detect stimuli originating from within the body. And then proprioceptors detect changes in movements, equilibrium, or body position. So the general sensory receptors are classified structurally as free or encapsulated nerve endings of sensory neurons. The free endings are mainly receptors for temperature and pain, with two exceptions. The epithelial tactile complexes detect light pressure, and hair follicle receptors respond to hair deflection. Again, these are the free endings. And then the encapsulated endings, which are mechanoreceptors, include tactile corpuscles, lamellar corpuscles, bulbous corpuscles, muscle spindles, tendon organs, and joint kinesthetic receptors. I got all of this from the notes that the professors put on their Canvas course. So if you don't have access to this, let me know and I can email it to you. But it's basically just like this table where they break it all down. And it tells you the location, the stimulus, and then where it's found in the body. Because I know this is a lot of words and yeah. Okay. And then one of the last things are the cranial nerves. So we are going to talk about this in lab. My class hasn't gotten there yet, but for lecture, I think you're going to need to know what specific cranial nerves have to do with what sense in your body. So we have 12 cranial nerves on each side. So we have a total of 24. And to me, the best way to learn it is to learn the names first and then learn the mnemonic for it. So there's like a million mnemonics online. You can find one that works for you. The one that I didn't come up with this one. This one was on, I think it was in the book. It's on occasion, our trusty truck acts funny. Very good vehicle anyhow. So again, that may not stick with you. There's some that are funny or not very appropriate online. So if you can find one that works for you, do that. The way that I learned it was, again, memorizing them in order because they go in order. So like 1 is olfactory, it's here. 2 is optic, and it's here. So they go down in order, learning the names of them first, and then the Roman numerals, and then learning the actual mnemonic for it. There is another mnemonic for their function, whether they are involved in the sensory, motor, or both. So that one, and it also goes in order. So let's say olfactory, smell, some, so it's for the sensory. It's some say marry money, but my brother says big brains matter more. Sensory number 1, olfactory. Sensory number 2, optic. Motor number 3, oculomotor. When we get to the B's, that means both. So that's 1, 2, 3, 4, 5 should be trigeminal. Would be both sensory and motor. So this one is good to know if whether in lab or on the lecture exam, they'll ask you cranial nerve number 7, facial, is involved in the sensory, motor, or both. Let's see, 1, 2, 3, 4, 5, 6, 7. So it would be both. Again, there's a lot of these online. This one to me sticks in my brain more. And then let's go through the cranial nerves and their function. So olfactory, smell, again, that's up here. This first one, the optic nerve, vision. 3, oculomotor. Let's follow it down. That is eye movement. Trochlear, number 4, vertical eye movement. Trigeminal, number 5, face sensation and chewing. 6, abducens is eye movement. 7, facial nerve, face movement and taste. Vestibulocochlear, hearing and balance, that's number 8. Number 9, glossopharyngeal nerves, throat sensation, taste, and swallowing. 10 is the vagus or vagus nerve, movement, sensation, and the abdominal organs. What was I saying? 11 is neck movement. And then 12, hypoglossal should be tongue movement. Yes, number 12, tongue movement. So again, they go in order as you see them, 1 through 12. There's 24 of them total. And then you need to know, actually, I need to ask my professor, but I'm pretty sure you're going to want to know the actual name of them and not just the Roman numeral. I know some professors accept the Roman numeral, but it'd be best to just learn the whole name of it. What else? Again, the mnemonic for this one, on occasion, our trusty truck acts funny. Very good vehicle, anyhow. Okay. And then I know we just learned them, but I do have a quick game. Oh, I thought this was the cranial nerves. It's the specific parts of the brain and their functions. So I'm going to give you a few minutes to kind of look at them if you can read them, and then we'll go over them. Oh, and then if anybody in the chat has any questions or comments, you can leave them now because it's the end of our PowerPoint. Oh, just kidding, no one's there anymore. Okay, we're going to post this on YouTube. If you're seeing this before your exam, we have practice exams in the Science Learning Center, FRIO 111.
Was there someone there?
There was 2 people.
Hello?
Mhm.